Hello students, this is Kanika. Welcome to my channel Prep Biology. So this is class 14th of this unit. Today we are going to discuss about very important topic of this unit, that is post fertilization events. Post fertilization events means the events which occurs after the fertilization. Okay, so in the previous lecture we have discussed the double fertilization. There the two structures are formed, that is the endosperm and the zygote. Now under the post fertilization we have the events. First is the endosperm development, second embryo development, seed maturation, fruit, formation of the fruit. Now the events of the endosperm and the embryo development and maturation of the seeds from ovule and maturation of ovary to form the fruit. These all events comes under the post fertilization events. Okay, so one by one we will discuss in this class. So in this class we will discuss about first two developments. First is the endosperm development and second is the embryo development. Okay, and in the next class we will discuss about the seed and the fruit. Okay, so let's start with the lecture. If we see the first event under the post fertilization event is the endosperm. How endosperm develop? Okay, now we know that double fertilization contains two fertilization. First is the male gamete fused with the egg cell, syngamy occurs and the formation of zygote. Second fertilization is male gametes fused with the polar nuclei. That fusion is called the triple fusion and the endosperm development. Now let's talk about the endosperm. When endosperm developed, in the previous class we have discussed about the primary endosperm cell, which is also called the PEC. Now the PEC will rapidly divide and form the triploid endosperm tissue. Okay, so the PEC, primary endosperm cell, will rapidly divide and form the triploid because its polarity is 3n tissues. This cell develop into the triploid endosperm tissues. Now if we see that these triploid endosperm tissues are filled with the nutrition, they are filled with the food material, which give nutrition to the embryo. Okay, so the endosperm tissues which are formed from the endosperm cell, that is PEC, they are filled with the food reserve and give the nutrition to the embryo. Now if we see that if the nucleus of each endosperm will divide or the successive division occur, these are the nucleus, and the nucleus of each endosperm will divide or rapidly divide, they will form the free nuclear endosperm. Okay, so the nucleus of the endosperm, they will divide the free nuclear divisions and those nucleus are called those endosperms are called the free nuclear endosperm. Okay, but if we see these nucleus when these nucleus will form cell wall around it, these nucleus individually will form cell wall around it, they are called the endosperm. They are called the endosperm, cellular endosperm, cellular endosperm. Okay, so these 2 points are very, I hope these 2 points are clear to you all. First, these endospermic tissues, if the nucleus of these endospermic tissues, they are divide rapidly, that is the free nuclear division occurs, these endosperms are called the free nuclear endosperm. Okay, but if when these nucleus form the cell wall around it, this is the cell wall around it, that endosperm is called the cellular endosperm. Okay, this is the nuclear endosperm without cell wall, and if the cell wall formed around the nucleus, that endosperm is called the cellular endosperm. Okay, I hope these two terms are clear to you all. Now, if we see the examples, under the endosperm, first example is the endosperm completely consumed during the embryo development before maturation. If in case when the maturation occurs, the complete endosperm is consumed by the embryo. If during the embryo development the endosperm is completely consumed, why completely consumed? Because endosperm will give the nutrition or food to the embryo development during the embryo development. Okay, when the endosperm completely consumed during the embryo development before maturation, this kind of situation occurs in pea plant, groundnut, the beans. Okay, these examples are very important. Okay, second type of the example is if they persist in a mature seed, if they are not completely consumed during the embryo development, they persist in the mature seed. This kind of situation occurs in castor plant and the coconut plant. Okay, so these examples are very important. If we see the coconut, if this is the coconut, and inside it we know the coconut water is present, and around it we know the white kernel is present. Okay, white portion is present, which we are usually eat. Okay, and coconut water is consumed by many of us. Okay, so if we see this coconut water is basically a free nuclear endosperm. That is a nuclear endosperm. Okay, so coconut water, if we see the coconut water inside the coconut, that is basically a free nuclear endosperm. Okay, this is made up of around thousands of the nuclei. Okay, and if we see the whitish part, that is the white kernel, that is basically the cellular endosperm. Okay, so what is cellular endosperm? We have discussed cellular endosperm when the nuclei form the cell wall around it. Okay, so that endosperm is called the cellular endosperm. Okay, the white kernel is cellular endosperm, and the coconut water is basically a free nuclear endosperm. This is all about the endosperm development or the different examples under it. Okay, now the second most important event under the post fertilization is the embryo development. We have studied the endosperm development. Now we are going to discuss about the embryo development. There are the different kinds of the embryo in different species of the plant. Okay, so we know that in the double fertilization, by the process of syngamy, the zygote will form. The ploidy of the zygote is 2n, diploid in nature. Now that zygote will develop and form the embryo. Okay, so the zygote will develop. This is the zygote, and the multiple divisions occurs and they will form the embryo. Now we are going to discuss about the different stages of the development of the embryo. Okay, so the early stages of the embryo development, okay, if the early stages of the embryo, there are some early stages of the embryo development. For that we will use the term that is the embryogeny. Okay, embryogeny means there are the different earlier development stages of the embryo development. Under the embryogeny early stages, we have different stages. First is the zygote. The zygote will develop into the proembryo. Proembryo will further develop, multiple changes occurs and form the globular stage. Fourth is the globular stage is converted into the heart-shaped stage, and the heart-shaped stage will mature into two kinds of embryo. First is in the monocot embryo, second is the dicot embryo. So these all are the stages, early stages of the embryo development. That is also called the embryogeny. Okay, so these all are the events that are going to occur during the embryo development. Okay, now if we see this picture, this is very important diagram of the different stages of the embryo. First, we know there is the zygote. Okay, this is the zygote. If you can see here, this is the zygote. This is the zygote. Now, the zygote will divide further and forms the proembryo. Proembryo. Okay, proembryo means the structure which is formed before the embryo. Okay, now this proembryo will develop into the globular embryo. As you all can see, this is the globular structure of the embryo. So this is the globular embryo. Now after the globular embryo, which stage is there? Heart-shaped embryo. As you all can see, this is the heart-shaped embryo. Heart-shaped embryo. Now this is the proper mature embryo. These all are the stages that comes under the embryo development. If you all can see here, the zygote will divide and it will form the 6 to 10 series of cells which is filamentous in nature. That part is called the suspensor. This is very important part. This is also suspensor, this is also suspensor. The function of the suspensor is it will join the zygote with the globular embryo. Okay, it will join the zygote with the endosperm. Okay, so the function of the suspensor, it will join the embryo with the neighboring tissues. Okay, so this is the suspensor. Now after the globular stage, you all can see here, this is the heart-shaped embryo. Okay, this is the heart-shaped embryo. The structure of the embryo look like the heart. That's why it is shaped. This embryo is called the heart-shaped embryo. Okay, after the heart-shaped embryo, the embryo will mature. Okay, it will form cotyledons. If they are formed the single cotyledon, that is called the monocot embryo. If it is formed the two cotyledons. This is the dicot embryo. Okay, you know that the cotyledons, what is the function of the cotyledon? The cotyledon will store the food. Okay, it will store the food which helps in the development. I hope these all stages, this picture is clear to you all. This diagram is very, very important for your board exam point of view. So practice this diagram, and these all parts are also very important with their function. Okay. Now, if we talk about the types of the embryo, first we will discuss about the dicot embryo. Second, we will discuss about the monocot embryo. As the name suggests, dicot means it is made up of dicotyledons. Di means two, cotyledons, having the two cotyledons. Okay. Now look at this structure. This is the structure of the dicot embryo. If this is the dicot embryo, we are going to draw the embryonal axis of it. Okay. So this is the embryonal axis. Okay. So the embryonal axis will decide that if the portion of the embryonal axis, the portion below the level of the cotyledon, if this is one cotyledon and this is second cotyledon, and from here they will originate. Okay, so below the level of the cotyledon, the portion is called the hypocotyle, and above the level of the cotyledon, that is called the epicotyle. I hope these two terms are clear to you all. Above the level of the cotyledons, that part is called the epicotyle, and below the level of the cotyledons, that part is called the hypocotyle. Now, what is the function of both of it? The hypocotyle first. Hypocotyle, that is the below. Hypocotyle will form the radical part, and radical will form the root cap. Okay, so hypocotyle will form the radical. Here the radical is present, and radical is the part that forms the roots. R for radical, R for roots, and the roots are covered by the root cap. This is the root cap. Okay, so hypocotyle will form the radical, and radical will form the roots, and the covering of the root is called the root cap. Now, if we see the epicotyle part, which is above the level of the cotyledon, the epicotyle will form the plumule. This is very important. Plumule. Okay, plumule is the part from where the shoot arises, shoot system formed. Okay, plumule. Okay, and the plumule will form the shoot system. So I hope this is all clear to you all. Okay, and this is the first cotyledon, and this is the cotyledon, and this is the second cotyledon. Okay, so this is all about the dicot embryo. Why dicot embryo? Because it contains two cotyledons. Okay, and these all parts are very important. What is the function of hypocotyle? Hypocotyle forms the radical part. Radical is the part from where the roots arises. So the covering of the root is root cap. Epicotyle forms the plumule part. Plumule will form the shoot system. Okay, now if we talk about the monocot embryo. In the monocot embryo, we have firstly draw the embryonal axis. This is the embryonal axis. Okay, This is the embryonal axis. Now in this picture, look at this picture very carefully. So this part, from this part, this is this cotyledon arises. Okay, so this is the cotyledon part. This is the cotyledon part, and from this part, the cotyledon arises. That's why I have drawn the embryonal axis like this. If we see the embryonal axis, same as we have studied in the dicot, below the level of the embryonal axis, below the level of the cotyledon, that is called the hypocotyle, and above the level of the cotyledon, that is called the epicotyle. This is same. Okay, now this is the hypocotyle. Okay, now hypocotyle will form the radicle that forms the roots. Okay, now here one very important thing is the covering of the radicle. The sheath of the radicle is called the coleorhiza. Okay, this term is very important. What is coleorhiza? The sheath of the radicle or the covering of the radicle is called the coleorhiza. Okay, so if we see in this picture, so this is the radicle. Okay, and the covering, this is the covering. This is the covering that is called the coleorhiza. Okay, you can learn with the, this is, in this term R is given, R for radical. Okay, so you can learn by this trick that coleorhiza has the R for the radical. Okay, the sheath of the radical is the coleorhiza. Now if we see the epicotyle will form the plumule. Now plumule will form the shoot. The covering of the plumule is called the coleoptile. Okay, the covering of the plumule is called the coleoptile. So you can learn with this trick. P is given, so P for plumule. Okay, so the sheath of the plumule or the covering of the plumule is coleoptile, and the covering of the radicle is called the coleorhiza. Okay, now here is very important part, that is the differentiated part from the dicot embryo, that this is the part which is present laterally or one side of the embryo. Okay, so this is the part which is called the scutellum. This part is very important. What is scutellum? Scutellum is the single cotyledon. It is the shield or single cotyledon which is present one side of embryo or laterally present. So this is the embryo, and this is present laterally or one side of the embryo. Okay, which is present one side of the embryo. I hope this is clear to you all. Scutellum is a shield which is the single cotyledon, okay, present laterally or the one side of the embryo. I hope these all parts are clear to you all. Now, if we compile all the events, under the post-fertilization, we have discussed the endosperm, we have discussed the embryo. Okay, in this class, this is part 1 of post-fertilization events. Now, in the next part, in the next lecture, we will discuss about the seed and fruit. Okay, these also comes under the post-fertilization events. I hope this lecture is clear to you all. If you like this lecture, like, share and subscribe my channel, that is Prep Biology. Keep studying. Study hard, study smart. Thank you so much.